Our next presentation is Prime Medicine, and I'd like to welcome Mohammed Asmal, Senior Vice President, Head of Clinical, to the stage.
Good morning. Thank you to the organizers for inviting us to speak. There we go. All right, it's my pleasure to introduce you all to Prime Medicine, a clinical stage gene editing platform company. We have had some major announcements recently in regards to a focused pipeline and a major collaboration with Bristol Myers Squibb. Today, I will walk you through some data and our strategy, which we believe leave us in a prime position to deliver on the promise of personalized gene editing to individuals with ultra-rare, rare, and common severe diseases. In a dynamic regulatory environment and with delivery technologies advancing as they are, we believe now is the time for a truly personalized approach with a differentiated safety profile. A little bit about the company, founded in 2019 out of the seminal work of Dr. David Liu and Andrew Anzalone, based in Cambridge, Massachusetts, with about 200 plus employees, all dedicated to building out our core competencies in RNA technologies, in artificial intelligence and machine learning in order to leave us with a solid foundation for launching into the future. What is prime editing? So prime editing, in 2 words, is precision and versatility. Prime editing couples the precision of the CRISPR-Cas9 targeting system along with the versatility of a reverse transcriptase, allowing the prime editing complex to be able to address approximately 90% of all mutations responsible for human genetic diseases, including transitions, transversions, small deletions, and small insertions. In addition, prime editing can be used to excise large chunks of repeat nucleotides that cause repeat expansion diseases. Finally, with the insertion of a recognition sequence and pairing with an integrase, prime editing can be used to integrate large chunks of DNA into the genome in a process that we call passage. We have established a broad portfolio of intellectual property around the initial publication of Dr. Anzalone and Dr. Liu and extended that beyond to include multiple different configurations of the enzymes as well as multiple different nucleic acid configurations. As I began this talk, I noted that we recently announced a restructuring of our pipeline, and this is what it looks like as of about a week ago. Our pipeline is spearheaded by our hematology and immuno-oncology programs, and in particular, the chronic granulomatous program, chronic granulomatous disease program, which is poised to deliver clinical data in the upcoming year. We've also announced, as I mentioned, a major collaboration with Bristol Myers Squibb to develop augmented CAR T-cell therapies. We believe that with Prime scientists' expertise in manipulating the genome and Bristol Myers Squibb's long history of working in immuno-oncology, we will be able to accelerate the development of novel CAR T-cell technologies. Next to the clinic is our program with a liver-targeted lipid nanoparticle for Wilson's disease. We believe that this modular lipid nanoparticle configuration, once demonstrated in Wilson's disease, can be expanded to include a large host of other common and rare diseases. Finally, we have also disclosed a collaboration with the Cystic Fibrosis Foundation to work at addressing the high unmet need for individuals who are not currently served by existing therapies. Importantly, our prior programs we have advanced to late preclinical stages, and so we have advanced preclinical data in the neurology, hearing loss, and vision loss therapeutic areas that are ready to be partnered out or to be advanced internally should the opportunity present itself. Our lead program to the clinic is in chronic granulomatous disease with our product PM359. This is an ex vivo autologous hematopoietic stem cell therapy, the likes of which have been documented through numerous clinical trials to be highly efficacious in other diseases. Chronic granulomatous disease is caused by mutations in any one of several enzymes that comprise the NADPH oxidase complex. This complex is important for the destruction of consumed pathogens in phagocytes. PM359 targets the NCF1 del-GT mutation, which results in a defective P47 protein. This is the most common cause of autosomal recessive chronic granulomatous disease, CGD. Patients with CGD, because of their inability to destroy pathogens, suffer from severe life-threatening infections, as well as autoimmune complications. They have progressive organ dysfunction throughout their life and have truncated morbidity and elevated mortality, often not living beyond the age of 40 or 50. The only curative option is allogeneic stem cell transplant, which is reserved for individuals with a matched donor and who are young, and it comes with significant attendant risks. We have assembled a significant data package in humanized immunodeficient mice to support the probability of success of PM359 in individuals with NCF1 mutated chronic granulomatous disease. On the left, you can see that mice transplanted with human cells that have modified by prime editing have the same engraftment capacity as unmodified cells. In the middle panel, you can see that these cells continue to express a high level of editing many weeks to months after they have been engrafted in the mice. The level of editing exceeding 80% is substantially above the anticipated threshold for clinical benefit, which is at 20%. And on the far right, you can see that the human neutrophils that have been modified by prime editing have restoration of NADPH oxidase activity, again, exceeding 80% of healthy volunteers, which is far above the 20% threshold that we believe is necessary for therapeutic benefit. In addition to having demonstrated the efficacy of PM359, we've also done extensive off-target and genomic analyses to establish the safety of this therapeutic product. So on the left, you can see that we have assessed 550 different in silico identified genomic loci for in vitro off-target activity and have seen none. In the right two panels, you see a comparison between a prime editor and basic CRISPR-Cas9, both targeting the exact same locus in NCF1. On the left, you see that there is no genomic damage that we could identify caused by the prime editor, so no deletions or translocations at that locus. In contrast, if you just apply basic CRISPR-Cas9, lacking the same control mechanisms and proofreading mechanisms that prime editing has, you have a 10% translocation risk. PM359 is, as I mentioned, our lead clinical program. So this is a diagram of what our clinical development plan is anticipated to be. As I mentioned, PM359 is an autologous hematopoietic stem cell product. This means that it requires a lengthy manufacturing and testing procedure before it can be administered to patients. However, because of the nature of the disease that we are studying and the nature of the autologous stem cell transplant product, we anticipate having a very near-term safety and efficacy readout. Because neutrophils are the primary cells that we will be measuring for both safety and efficacy, and these are the first cells to engraft, we will be able to determine both the safety in terms of engraftment potential and the efficacy in terms of reconstitution of NADPH oxidase activity in a very short time frame, potentially a few months, after treatment. And hence, we anticipate initial clinical results next year. Furthermore, we have structured the study such that we rapidly age de-escalate from adults to pediatric patients. This is because the high unmet need for this disease really is in the pediatric population, as exemplified by the fact that these patients are offered allogeneic transplant if they have matched donors. In addition, based on the precedent in this field, we hope that with promising early clinical data in our first-in-human trial, to rapidly expand this into a pivotal trial. Our second program to enter the clinic will be in Wilson's disease. So Wilson's disease is a disease of copper transport due to mutations in the ATP7B gene. It's autosomal recessive, but it also happens to be one of the most common rare diseases at a prevalence of approximately 1 in 30,000. It causes progressive lymphodamage as well as has neuropsychiatric symptoms in individuals that have been affected. Although copper chelation is available standard of care, problems with lifetime adherence as well as side effects do result in a significant unmet need. This is some initial data. This is actually pretty old, outdated data showing the efficacy of our lipid nanoparticle therapy for Wilson's disease. So on the left two panels, you see in vitro ability to correct one of the 2 prevalent mutations in Wilson's disease. On the right, you see data from another program, which we have temporarily paused, which we see in vivo NHP surrogate data demonstrating ability to achieve over 60% gene editing in non-human primates with this. We believe that this modular platform will allow us to address a large swath of unmet need in liver diseases. Please stay tuned for updated data coming up in the next couple of months in major conferences. Finally, I'd like to shift gears a little bit and talk a little bit about the data that has gotten external partners excited about working with Prime Medicine. First off, this is the data that supports our collaboration with BMS. This has been generated by Prime scientists. So on the left, you see the ability to integrate with high efficiency a CAR construct precisely at the TCR locus in T cells. This allows an unprecedented level of safety as well as regulated modulation, not possible with current LVV manufacturing techniques. In the middle panel, you see the ability to multiplex CAR integration along with other modifications with very high efficiency, such that we can generate either allogeneic or augmented T cells with unprecedented ability to avoid double-strand breaks. And on the right, you see the ability of these modified CAR T cells, prime-modified CAR T cells, to kill cancer cells in vitro. Oops. Oh, sorry. Finally, some data that supports our program, our partnership moving forward in cystic fibrosis. So Prime Medicine is dedicated to both addressing the unmet need for mutations, about 10% of all cystic fibrosis mutations that are not currently addressed by the available therapeutics, small molecule therapeutics that are out there. In addition, Prime Medicine is also dedicated to be able to provide durable treatment for those individuals with common mutations. So what you see on the left is in vitro, very high efficiency correction of one of those unserved mutations, as well as correction of the most common mutation for causing severe CF, the DEL508 mutation. On the right, you see phenotypic correction in patient-derived intestinal organoid cells, in which we were able to reverse cell swelling phenotype in intestinal organoids using prime editing, and that was not possible with an available therapeutic. So finally, in closing, this is Sebastian. He is a young man with chronic granulomatous disease. He was lucky. He got an allogeneic transplant when he was approximately 2 years old. It was a very fraught procedure. He was in the hospital for almost a year. We're hoping to bring to individuals like Sebastian and who have other severe diseases a personalized medicine approach without many of the safety concerns that are attendant to current available therapeutics. Thank you very much.